Good afternoon, everyone. Dr. Qinglan and myself are extremely honored to speak today at the symposium honoring Dr. Fraumeni, and we'll be talking about indoor air pollution and lung cancer in China. As Bill just mentioned, Chairman Mao and President Nixon met in February 1972 and opened up the door to a whole series of scientific collaborations in China. We fast forward to 1982, and you can see both Bill Blott and Dr. Fraumeni and his wife, and they literally leaped through that door and set up an extraordinary research program that has gone on since that time, is multifaceted, covers many exposures and many tumors. Now, there were many reasons to set up a cancer research program in China. There were extraordinary opportunities with regard to different types of diseases that presented in different ways, different rates, different exposures driving those diseases. This is one example of why it was compelling to study lung cancer in China. And here you see a wide spectrum, wide variety of rates, lung cancer incidence rates among women in selected Asian countries with very low rates of female smoking. This is work that Michael Toom published, and you can see he's comparing rates off to the left among women in Connecticut, and that's 1935 to 1940 before women began smoking. And you can see a wide variation in rates, very high rates in China, and geographic variation in those rates. So, as Bill said, this was a sign that there's something interesting going on, something that deserved our study. So Dr. Fraumeni and Dr. Blott, along with their collaborator, published a paper noting the geographic variation of female lung cancer in China in 1986. And you can see here, even within China, even though rates overall were elevated, a strong variation from rates, some hotspots up in the northeast and also down in the southwest and also around Shanghai. There was more than a 20-fold variation in lung cancer rates in the population. This led to a series of studies that Dr. Fraumeni and his colleagues conducted. Dr. Fraumeni was the lead investigator in a number of these and supported other work. There were 4 primary areas of study that he worked on: Shanghai, Gansu, Shenyang, and Xuanwei. Shanghai was a very large population-based study that Dr. Fraumeni conducted with Gao Yutang and Wei Zhang, who's here today. And one of the most notable findings was an increased risk associated with exposure to cooking fumes from oil that was heated to high temperatures, particularly a certain type of oil called rapeseed oil. And there was some wonderful mechanistic work that followed up on that, conducted by Peter Shields and Kurt Harris, that showed a very high mutagenic activity to rapeseed oil. This finding was replicated in a case control study in Gansu, where individuals lived in homes that were actually underground, had very poor ventilation, and work that was led by Jay Lubin. The finding was replicated. This was also a study that, with Jay and John Boyce, identified some important dose-response relationships with indoor radon and lung cancer. But what I'm going to talk about today are studies in Shenyang and Xuanwei, where the focus was in particular on coal. Now, 800 million people worldwide use coal for home cooking and heating still today. And coal is used to generate electricity in many countries. So this was a very important study that was conducted, very large for its time, a population-based study of lung cancer in Shenyang, in the very northern part of China. And one of the exposures that they focused on was exposure to something called a keng. A keng is a very ingenious device that cooks coal, burns coal here, uses it for cooking, and then the heat is channeled through the bed and keeps it warm. It's very cold. This is what an actual keng looks like. There's the stove, there's the funneling underneath the bed. And they found a strong dose-response relationship between burning coal and a keng for women, not men. And then when they looked at burning a keng in the home and actually in the bedroom, so they were both cooking in the bedroom as well as burning fuel in the bedroom for heating, they found a dose-response relationship for both men and women. And in contrast, there was no association with lung cancer risk for people who were using central heating that was supplied by gas. Now, this was followed up in a collaboration between Dr. Fraumeni and Dr. Henderson, where the female cases and controls from the Shenyang study were combined with female cases and controls from a study in Harbin, relatively close by, also in the northeast, very cold, and almost 1,000 cases and 1,000 controls, this time of women. And again, they found a dose-response relationship for burning a keng, particularly in the bedroom, for using coal stoves, but not for using non-coal stoves. So this was very important evidence that burning coal for heating and cooking inside the home was associated with lung cancer. Now, another region of China that Dr. Fraumeni was very interested in pursuing research on was in Xuanwei. Xuanwei is located in the southwest part of China, in Yunnan province, and rates there among the very highest in China. In fact, they may be among the very, very highest in the world. There are a number of different types of coal that are burned in Xuanwei. Something called locally smoky coal, or a variation on bituminous coal, smokeless coal, a type of anthracite coal, and wood. And unlike in the Northeast, this area is much poorer, so people just dig a hole, make a fire pit in the home, and then burn the coal there in homes that were generally unventilated and got very high exposures. Now, Xuanwei is a semi-mountainous rural region. It's actually relatively cold, even though it's in the south, because it's fairly elevated. Almost no women smoke. Most of the men smoke, though at light levels. And it's really a model and a stable population to study. There was only one problem, and that is that the US EPA got there before NCI. The US EPA had a team that was doing research in the late 80s, and it wasn't quite an opportunity to work there. But Dr. Fraumeni bided his time. He waited till 1999, and then instead of having to go to Xuanwei, Xuanwei came to him. And came to him in the form of Dr. Qing Lan. Dr. Fraumeni recruited Qing in 1999, just after she had finished her PhD at Chapel Hill in a joint training program with the Chinese Academy of Preventive Medicine in Beijing. And Qing, for her dissertation, had established a very large cohort in Xuanwei, the goal of the cohort was to study the causes of lung cancer excess and also test if lung cancer mortality decreased after installing stoves with chimneys. The Chinese government, in recognition of the potential problem of people being exposed to very high levels of coal combustion products, had given a few dollars, made available a few dollars to each home to replace their fire pits with a stove and a chimney. And there was an option whether to use it or not. Not everybody did. So Qing came here in 1999, brought the entire cohort data set with her, and also brought thousands of samples from a pilot molecular epidemiology study that she had done. The investigators from the EPA had retired and agreed to transfer the whole study to NCI. So Dr. Fraumeni had the opportunity finally to work in Xuanwei, and he worked with Qing to produce some really exciting results. So one of the initial observations is that there's a striking increased risk of lung cancer in men and women associated with exposure to smoky coal. In women, the risk was almost 100-fold. It was also quite interesting that the age started cooking was strongly associated with risk of lung cancer among women using smoky coal. And you can see the peak risk starts among women who started cooking between the ages of 12 to 15. No men cooked in that population. Equally important was a striking observation that Qing made in a paper that she published, showing that improved stove ventilation substantially reduced the risk of lung cancer. Almost a decrease of almost 60%, of almost 40% in both men and women. This is one of the very first examples of proof that an intervention, such as improving ventilation in the homes with very high levels of indoor air pollution, actually works, that actually reduces lung cancer. And her group also showed that the same intervention had a strong influence in reducing the risk of COPD. Now, there are a series of ongoing projects taking place in Xuanwei now, trying to evaluate the causative agents in smoky coal and lung cancer, and to get at the underlying mechanisms of this association. There are studies going on looking at coal composition, coal-controlled combustion studies, There's a great deal of air and personal air monitoring exposure assessment that's going on. Mechanistic studies are taking place with the goal to link all of this to lung cancer risk. This shows some initial data that we've collected, personal air monitoring of non-smoking women in this region in China. And if you look at smoky coal, you can see that, yes, it seems like there are very high levels of pHs. Interestingly, moderate levels of respirable quartz, PM 2.5. But then you look at what we detected in smokeless coal and in wood, and you see, well, there's some pHs in wood, some respirable quartz, more PM 2.5 and higher levels of SO2 in smokeless coal than either. So simply analyzing the coal isn't adequate to try to get a handle on what's driving this extraordinary excess. In parallel, we're conducting some mechanistic studies to try to get at this question biologically. This is work that Qing set up through a collaboration with Avi Spira at Boston University. And here, what we've detected is a 282 gene signature in this gene expression profile that reflects a response to smoky versus smokeless coal. And you can see some genes are increased, some are decreased. These are individuals with smoky coal exposure. These are individuals with smokeless coal, the low-risk coal exposure. We then tried to take the exposures that had been measured on these individuals and tried to look at the relationship between those air monitoring levels and this gene expression signature. And what we found was that the very carcinogenic pHs, particularly dibenzanthracene and the marker benzoate showed a correlation, significant correlation with this gene signature, but non-carcinogenic pHs, and interestingly, PM 2.5 showed no association. We also found that the changes induced in smoky versus smokeless coal exposure were similar, showed enrichment in some of the same genes that have been identified that distinguish the response in current versus never smoking. Now, the impact of this research was quite important. Dr. Fraumeni's study in Shenyang and Qing's study in Xuanwei were the key contributors to IARC's decision to conclude that coal, indoor air emissions from coal are group 1 carcinogenic compounds. And this was affirmed in 2009. In addition, these findings played an important role in the development of new WHO guidelines for indoor air quality. These were adopted just last week, and they promise to, if implemented, make an important contribution to reducing the substantial morbidity and mortality from indoor air pollution throughout the developing world. Now, the next steps in the Xuanwei study will be the integration of exposure data into the analysis of lung cancer and a new case control study, histologic and tumor marker characterization and integration into cancer risk analysis, and the comparison of findings with lung cancer in other parts of China and Asia and the West. And along these lines, Qing established a new consortium, the Female Lung Cancer Consortium in Asia. And the goal of this first notes that lung cancer in Asia, as I noted before, it takes place in women, mostly among never smokers, that the rates are relatively high compared to the West. Interestingly, a higher proportion is an adenocarcinoma, and it has distinct molecular characteristics with clinical ramifications. The genetic and environmental risk factors for lung cancer may differ among women in Asia versus the West. This is one of the driving hypotheses in the consortium, and the goal of the consortium is to study both the environmental and genetic etiology in this population. This was the first project that the consortium conducted, a large-scale GWAS, the largest ever of never-smoking females, and this was done in a collaboration between Qing and Stephen Chanik and many collaborators in Asia. And at NCI, there were 18 studies that were involved, 6,600 never-smoking female lung cancer cases, mostly adenocarcinoma, and 7,500 controls. And we found 3 novel gene regions. One of these 3 was particularly interesting in ROS1, since ROS1 fusions in lung adenocarcinoma are considered to be one of the drivers of oncogenesis. Also, what was quite interesting was to look at findings that had been detected in smoking Caucasians that had been associated with risk of lung cancer. And we saw absolutely no association among Asian never-smoking females. This was a follow-up of a previous report that Dr. Margaret Spitz had published And we see evidence that this locus, these associations, really do seem to be specific to smoking and lung cancer, that in this population there was no relationship. And then finally, initial analysis to study potential gene-environment interactions in this GWAS has identified several, in fact 3 of the 7 SNPs that showed association provide evidence of interaction. This is being followed up now. Now, if we take a step back and look beyond China and beyond just coal, we should note that half of the world's population is exposed to smoke from cooking and heating with solid fuels. In addition to coal, that includes wood, plants, and dung. This is a major risk factor for global disease burden, but cancer risk for non-coal fuel is unresolved. And there are an estimated 3.9 million premature deaths annually. So the future course of this consortium is to expand it to include studies in populations exposed to biomass in Southeast and West Asia, to define a spectrum of susceptibility alleles and search for additional interactions, and very importantly, to conduct tumor marker characterization in adenocarcinoma samples using sequencing in particular from diverse populations. This shows the expanded consortium and includes studies from Japan to Mumbai, India, from Singapore up to Beijing and Shenyang, and offers a striking opportunity to compare the epidemiologic exposure data, genetic data, and ultimately tumor data across these diverse populations. Before I finish, I want to touch just a little bit about occupational studies. Dr. Blott described important work that Dr. Fraumeni and his colleagues had conducted in the United States on occupation. This is the same smokestack. I just wanted to note that this is a paper from another Lee, Anna Lee and Dr. Fraumeni. It was published in 1969 on risks of lung cancer among copper smelters. And I'd like to note that Dr. Fraumeni taught us to identify high-risk occupations, to set up high-risk populations, to set up high-quality case control or cohort studies to ensure that we have very high-quality exposure assessment, and that if we see associations and if we can replicate it, to not just stop there, to go on, and to attempt to then use the molecular tools in our armamentarium to try to understand underlying mechanisms because of the enrichment that molecular epidemiology approaches and epidemiology approaches have with each other and the synergy that can result from that work. These are a number of studies that resulted from that motivation that he provided us. Cohort studies of diesel exhaust conducted by Deborah Silverman in the U.S., of benzene, a large cohort study in the United States conducted by Martha Lynette and Bill Blott, a big study of formaldehyde that Aaron Blair and Laura Bean Friedman conducted in the U.S., and a series of mechanistic studies that Ching led to try to understand the underlying biologic basis of these associations. And this shows examples of how ideologic work can lay the foundation for public health action. These studies contribute importantly to IARC reviews. In all 3 instances, they provided either the key study, in some instances almost the only studies that we use, the epidemiologic studies that we use by IARC in their review and conclusions about the carcinogenicity of these compounds. And also, these have had an impact on regulation. They've had a public health impact. And we're particularly proud of some of the work that's resulted from our benzene studies. The benzene cohort study directly led to workplace reduction of benzene in China, and some molecular epidemiology studies that Ching conducted showing some important molecular changes in populations exposed to benzene at very low levels resulted in reducing the workplace standard in the Netherlands just last month to the lowest level in the world, and also directly led to the reduction of benzene in reformulated U.S. gasoline. I'd like to close with the Chinese version of "We Stand on the Shoulders of Giants." 河水不忘打金人. When you drink the well, when you drink the water, don't forget who dug the well. So Ching and I and our collaborators at NCI and the many collaborators that we have in China who Dr. Fraumeni developed long-standing collaborations with, who he helped to train, who he helped to mentor. In fact, there are multiple generations of Chinese investigators, many who came to NCI to be trained, have expressed to us over the last few weeks to make sure to tell him how grateful they are and how thankful they are for his mentorship and the work that he did to establish these international collaborations based upon mutual respect, based upon honesty, transparency, and that really launched decades and generations of studies that we are still harvesting today. Thank you.